So we'll begin our afternoon session. Our first speaker is Klages. He's got that long title.
Yeah, sorry for that. Thank you very much, and apologies for the long title. But first of all, I would like to thank very much the organizers, Luca and Anastasia, for giving me the opportunity. So this is actually the third time that I'm visiting INI since July last year, and the first two times was at the program that Luca and others organized. And really the talks that I gave there and preparing them, and then the feedback that I received from the people really shaped my thinking a lot. And indeed what I'm going to talk about now is in a way the third and final talk in this row, and you will see what I came up with. Okay, yeah, again, sorry for the title, but I just tried to put in, or to emphasize the application aspect a little bit. So as an outline, this is what I'm planning to talk about. Yeah, we're here open for business. So I will start with a more general motivation in terms of movement patterns, why one may be interested in them, and really a very basic outline of theory of stochastic processes for modeling movement. And then second part, I will actually, I'm actually a theoretical physicist by training, now also doing applied mathematics, but here I will really talk about 3 experiments. So here I will show results for experimental data analysis with the aim to construct data-driven stochastic models. For these 3 organisms, sea turtles, bumblebees, and cells. And the third part is then more again for the theorists. Namely, here I will try to put forward what I believe is a fairly general stochastic model in terms of kind of Newtonian-like dynamics related to Langevin dynamics in a co-moving frame, which I believe is quite promising for experimental applications and theoretical understanding. Okay, let's start with the motivation. Here you will see 3 pictures of 3 different organisms that kind of move. These are actually a type of white blood cells, neutrophil granulocytes, and they have all been put to the same origin, and they move on a chemical gradient parallel to the y-axis. So you see that, that there's actually an average drift along the y-axis, so they crawl, or they try to crawl along the chemical gradient, and as you can also see, they diffuse in both directions, so they spread. These are about 60 cells, and these are the mean positions after 30 minutes of where you find them. Second example is bumblebees. So here you have, this is actually a lab experiment, meaning you have an entrance here in front, and then you let in 1 bumblebee, and it forages in this artificial carpet of flowers where they can collect nectar. And again, you can track the trajectories of a bumblebee of how it searches in this environment. And the third one is the path of a sea turtle swimming in front of the coast of West Africa. This has been tagged by a sensor and could be tracked via GPS satellite. So I'm going to talk about all these 3 examples, but if you look at the path, there is perhaps one observation, namely they all kind of look random, right? Like random walks sort of thing, but in somewhat different ways. If you have a background in stochastic processes, you might wish to make an educated guess of what type of motion this might be, but it's perhaps not so obvious, right? But why, first of all, understanding these random-looking migration patterns? Because we're here for open for business applications, right? Actually, we heard the talk about cell migration this morning, so therefore for the cells I don't have to say too much. But here actually, certainly cells perform very different tasks in the body. For example, these are white blood cells, so it's immune defense. So you might be very interested to learn about the detailed properties of cells contributing to your body's immune defense. Also the game that physiologists or cell biologists like to play is, and in fact it was mentioned this morning, that they block certain molecular emitters by chemicals on a fine scale, and then checking whether this has any impact on the diffusive properties of a cell. And that was actually done by my colleagues, so we recorded different migration patterns for different types of cells, so wild types and ones which have been chemically modified. And that way you can also check for cell deficiencies, And perhaps you could even kind of build a kind of machinery to detect or to distinguish healthy ones from deficient ones. And that way, again, better understand what's going on in your body, whether you're ill, and so on and so forth. Now, a second one, actually I'm going to talk about foraging under predation risk. So that means bumblebees, if they search for nectar, may be killed by so-called crab spiders. So here you may wish to better understand the interactions of insects with the environment. Also for their conservation. And indeed this even more applies to the sea turtles because they actually belong to endangered species. So for wildlife conservation, you may wish to understand better their foraging activities. And even more, they have been tracked over many years. So in terms of climate change, for example, you could perhaps ask questions of whether their foraging behavior changes from year to year to check for the impact of climate change and things like that. So far to some motivation or why you might be interested to study migration patterns. Okay, now how do you analyze movement patterns? This goes in 3 stages. So first, obviously you have the experimental data and you want to do some statistical experimental data analysis. For example, here, this is a migrating cell. You record, you make photos under a microscope. Then you have the outline of the cell, you have to identify a cell center, then you have to construct a cell path, and then you have kind of to extract quantities characterizing your movement. And that already relates to the second stage, namely, that's at least what I'm interested in. I would like to construct stochastic models characterizing these movement paths. So that's what I'm interested in, that's what I'm after. And that means, of course, the type of stochastic model you want to construct kind of determines of how you look at the data, right? So because that requires certain data input in terms of, say, probability distributions, correlation functions. And that's how you analyze your data in the very first place, to extract these quantities from the data so that you can construct a model driven by the data. And then, of course, once you have a model, then you want to understand, okay, what's actually the mathematical, physical and biological properties. What does it mean, right? What does it tell me actually about the dynamics I started from? And then, of course, certainly the third level would be applications, right? Here's one example, actually. Whoops. So I may say that this is actually a movement path of a bumblebee that was constructed from a model. And this is the search problem. So you may have randomly distributed targets. And then you may ask questions about, say, search efficiency for a given stochastic process that you have constructed from data, meaning certain organisms should be interested to search efficiently to survive. And perhaps, you see here, perhaps you can even learn from organisms of how to search efficiently. And actually, this was exactly the title of a proposal that I submitted to a UK Research Council. And the title of the call was Nature Computes Better. And it was unfortunately rejected because it was deemed out of scope. I am not quite sure why, but proposal is still there. So as we here are open for business, nice three-page proposal. If anyone is interested, talk to me. So, but this is what you can do and what I'm also interested. So learning from organisms of how to search. And indeed, some of you may be familiar with the Levy hypothesis, which became quite famous. Right, so perhaps you can even learn from organisms how to search efficiently, and I will tell you later that maybe something more lurking in the background here. Okay, so but in this talk I will primarily focus on stages 1 and 2, so meaning data analysis and stochastic modeling and not so much 3. Okay, so let's start with some history. Carl Pearson, published a famous paper, a very short paper, just a few sentences essentially, in 1905, where he formulated the problem of the random walk. And here's one citation. Namely, he formulated the following problem. A man starts from a point O and walks L yards in a straight line. He then turns through any angle, whatever, and walks another L yards in a second straight line, and keeps going like that. And then he asks the question, okay, can anyone tell me about the probability to find the random walker at time t at a certain distance away from the origin? And the answer was, of course, given by Gaussian distributions as formulated by Lord Rayleigh. That coined the problem of the so-called random walk formulated by Pearson. So this is in a way a definition of the random walk in a plane. And in fact, that became famous as the drunken sailor problem, in the sense that here there is no memory really in the dynamics. All the steps and angles are uncorrelated. That's why Pearson called it the drunken sailor problem. In mathematical terms, of course, we have a Markov process here. And now, 1 year later, there was a second paper, again by Pearson, therein he proposed to actually model the migration of species by random walks. And again, I cite, the species scattering from a center is supposed to distribute itself uniformly in all directions with an average distance to which an individual of the species moves. So same idea of using a random walk for modeling the migration of organisms. So in that way, perhaps Pearson can even be considered as a kind of fauna of movement ecology in some abstract sense, I would say. So he's putting forward a simple random walk for modeling the movements of organisms here. And this was the first level. So the idea is to describe the movements of organisms by a simple random walk in the plane. But of course, this is just the very beginning. Then you see around the same time, Einstein formulated his theory of Brownian motion for a particle in a fluid. And this is a famous picture from an experiment by Perrin, who verified Einstein's theory a few years later. And around the same time, you see 1908, then certainly Langevin came up with this famous equation, which may be called Newton's law of stochastic physics, because that's essentially what it is. So you take Newton's second law. Well, the picture is not there anymore, unfortunately. It's now for selfies over there, but it's the same spirit that you have Newton's second law, and simply you formulate the force on the right-hand side a little bit differently. Namely, you split it into a viscous damping with Stokes friction and random kicks modeling the collisions with the surrounding fluid particles. Well, you all know that, I suppose, but this is just a brief reminder, because then there's something more to come about it. But this is the basic physical idea of writing down what's called a Langevin equation. Very straightforward, one-page paper. You just wrote it down based on physical intuition, and that's it. Mathematically, certainly, this is nothing else than an Ornstein-Uhlenbeck process for the velocity, which again is a Markov process. So far, so good. But now, again, from Brownian motion back to biological movements. So now we have the second level of more complicated random walk-like dynamics in terms of Langevin equations. And they have since Pearson, been widely applied to model biological dynamics. I just cite one paper by Skellum here, but there are plenty of others. So in a way, this works, especially by Pearson and then Langevin, really coined a whole paradigm that kind of dominated the movement of organisms for many, many decades. So use either random walks or Langevin dynamics to understand the movements of organisms. And here I mention a kind of link in between random walks and Langevin equations, which physicists call overdamped Langevin dynamics. So you go to the limit of high damping, so meaning you kind of ignore the acceleration on the left-hand side, and then you obtain what's called overdamped Langevin dynamics. So simply velocity is equal to noise. Where in this context, this is again Gaussian white noise. In mathematical terms, we are talking here about the Markovian Wiener process. So another player in the game. And indeed, I will focus on this one in the following very much. So mathematicians call this typically Brownian motion, while for physicists, the underdamped Langevin equation, that's what they call Brownian motion. Okay, now there comes a problem if you think of all this in view of modeling biological movements. Namely, the Langevin equation was certainly only formulated for the passive motion of a tracer particle in a fluid. Namely, a particle that doesn't move by itself, it's just randomly kicked by the collisions with the surrounding particles. But that's, of course, not of how an organism moves, right? An organism moves by itself. It's not passively driven by the surrounding particles. So there's a problem here. And that has not really been addressed by many people working in this field over many, many decades in the last century. They just took a random walk, and this Langevin equation, and still people are doing it, many are doing it, without addressing this problem of activity here. But because an organism certainly takes up energy from the environment, and converts it into kinetic energy. Again, we just heard about it this morning, right? A cell. The cell takes up ATP from the environment and converts it into movement. So you should build this into your model, otherwise there's something fundamentally missing here and it should have some consequences. And indeed, of course, this is not at all new because there's this big field now of active matter that emerged over the past few decades. Or here I specifically focus on the so-called active grounded particles. So what is this? Active grounded particles, well, so far I would say there are three generic examples that I'm going to discuss. First example, actually this is known since the 90s. So this is the Berlin school by Ebeling, Szymanski-Geyer, Schweitzer, and so on. You see many decades ago, they proposed already to model the active movements of a particle by this type of Langevin equation. So you have again Langevin dynamics. I don't care about mass here, but now the change is that you don't have constant Stokes friction. You make the friction velocity dependent. And here's an example. This is so-called Rayleigh friction. And this models exactly the take-up of energy from the environment and conversion into kinetic energy. Because that depends on whether your speed is larger or smaller than a given speed v0 by which an active particle wants to move. So either you take up energy or you lose energy. So this motion tries to stabilize around speed v0. Okay? And they published a lot of papers on this active particle model and also applied it to experiments. Okay, number two, another very popular active particle model is this one. Now we are back to overdamped Langevin dynamics, and now you see this is a kind of local drift, but the angle here is driven by rotational diffusion. So you have this Langevin equation, another one for the orientation angle, meaning this is the polar angle in the plane by which the particle moves. And you see this is obtained by a Wiener process on the circle. So this is yet another second active particle model by which you have external input by the fluctuation of the turning angle. Third one, this is now again overdamped Langevin equation, but here we have correlated noise. So this is typically Gaussian noise, but it's not white, it's correlated, exponentially correlated. So we have three different mechanisms to my knowledge. If anyone knows more, let me know. But these are three different generic mechanisms of how to generate active motion in terms of taking up energy and converting them into self-propelled motion in contrast to the movement of a passive particle. And they've been studied a lot in the literature. Actually, there's even a fourth one around. I mentioned here, namely this class of so-called run and tumble particles. Again, it was mentioned this morning. But I'm not going to address that here very much. I will focus on these three. And I may mention, as this is a meeting on anomalous diffusion, that all these three models exhibit normal diffusion at a long time limit. So here, per se, there is no mechanism of anomalies here. Okay, so far so good. Next, so this sets the scene. That's my introduction in a way. Rather basic recap of some known results. Now what I'm going to do is I will focus especially on the overdamped Langevin equation, but without any particular noise, so not assuming any particular functional form on the noise, but I will try to extract this equation from data. That's what I'm going to do. First example, loggerhead sea turtles, Caretta caretta. This is joint work with quite some colleagues who may even be following online. Hello if you are. Vijay Kumar, who is doing his PhD on that. Perla Torres, who did his PhD with Chris Isaac-Guia from Queen Mary in the biology department, and Vladimir Palgulin. So we have loggerhead sea turtle, this guy here, and they're pretty heavy. So they're about 1 meter long. You see the weight, about 125 kilos. They can be up to 80 years, and they can swim very amazingly fast, if up to 35 kilometers per hour, I found, but certainly only if they're threatened. And what was done in this experiment by Chris and Pella was that they attacked more than 20 turtles since 2011. So here you see a sensor, and then they tracked them with a satellite. And they were nesting on Cabo Verde Islands, close to West Africa, and then they were foraging close to the coastline. And the following results are for just one turtle, a female turtle that they called Mukamba. And just to give you an impression, 1,350 data points recorded over 400 days, 2012, 2013. We also had slightly larger data sets, say 2,200 data points over 3 years, but that's the kind of order of magnitude. And here we have a turtle pass. Okay, quality is not great, sorry for that. But here there is one example of a turtle pass, so that's the coastline. And yeah, again, sorry, it needs to be improved, but I hope you see a little bit what's going on. So you see the dots, which is where the positions were recorded. And now, if you look at it, there are 3 important observations. Namely, first of all, you see long gaps where nothing is happening, so there's just a line connecting them. So this is a fundamental problem if you observe movements in the wilds, namely that typically the time intervals are not regular. They are irregular because of, well, I mean, they dive when they cannot send out a signal, the satellite is not there, whatever, no? And then, of course, if you want to do statistical data analysis, you're facing a problem, right? You cannot just compute a PDF from irregularly sampled points. And my suspicion is that in quite some work, I mean, you have to be very careful. That's what I've learned. If you're not careful, you can get a mess. No? So it is a severe problem. Secondly, if you look at the data points, here is the scale. So we have colored them with the speed of how they swim. And you see that they do not swim homogeneously with, on average, the same speed or something like that. You see that here they're very slow, then they speed up. So I would say this is rather spatial temporal intermittency. So there are phases where they swim fast, there are phases where they search for food, where they're slow. And thirdly, well, look at this path. Okay, again, it's not great, but then make a guess of whether you would think this is Brownian motion. Right? And when I looked at paths like this, then I immediately saw, well, there are loops. There are loops all over the place, which look correlated. I come back to that. So there's looping. And again, you will see in a moment. And so first, what you now need to do is, of course, a lot of data cleaning. I don't go into detail, but this is highly non-trivial. And that's, I would never, ever just apply a blind machine learning onto this. It won't work, okay? So, I mean, you really have to be very, very careful. Only after the cleaning, you know what's going on. And here we did the most straightforward method, linear interpolation between data points. Okay, and now, as I said, our goal is to construct simply a stupid overdamped Langevin equation from the data, x dot equal to noise. What do you need? You need the PDF of the noise, and you need the velocity autocorrelation function. Very simple. And this is now, first, let's look at the speed distribution, which is related to the velocity distributions. And this is the data. And this is a fit with a Rayleigh distribution, a two-dimensional Maxwell-Boltzmann, which then corresponds to the Gaussians that you have for the velocities, Vx, Vy. And you see that it works pretty well overall. And this is a quantile plot, so it tells you that for small speeds, it actually works very well, while you even see it here for larger speeds, there are some deviations. And if you take a more careful look, you indeed find that there are exponential tails. Again, here QQ plot, and we tested on exponential in the power law. So we didn't get a Levy distribution, but instead, though the deviation is not great, but you see exponential is definitely a bit better than power law. So therefore, our conclusion is that the speed distribution is Rayleigh in the center with non-Rayleigh, meaning exponential tails. And indeed, overall, that fits very well to so-called gamma distribution, which actually has also been found for the famous albatross flights. Okay, and the same applies for velocity distributions. If you analyze them, again, you find in the center they are Gaussian with exponential tails. So you know about this, the velocity distributions. And then, of course, the second thing is the correlations. And my impression is really that kind of looking at correlations is still kind of undervalued in this field. More people should be doing it because in my view, but again, you can judge yourself by what I'm going to present, there's really very important information hidden in the velocity correlations. And this is an example. So here we define them by a time average along a trajectory because we cannot average over the ensemble of the turtles because they're all different. And this is what we get for the correlations along X and along Y. I need really to improve the figures. Again, sorry, but what's going on here is we have exponential decay on short time scales, so really just here. And then, big surprise, you see huge oscillations. Right? When we found that, I was kind of actually very happy because I suspected that because of the looping. There's relation to the looping, but I'm going to explain. But you see that actually, so here there are even regular oscillations with a certain period. So what you should see here is, yeah, barely seen, but there are data points, and then there's the fit with the red line. Yeah, your data you see, right? It's a bit more irregular, but you still see that the period is rather regular. And then we fit it with an exponential plus simply a cosine. And what surprised me a lot was that indeed these oscillations don't decay, they are persistent. So with a normal stochastic process, yeah, there's a question.
What's the time scale?
Hours.
Hours.
Yeah. So this is on very large time scales. I come back to that. So there are persistent oscillations that are best modeled like that. So you see even the data, they don't go away, they don't decay. Okay, and I would say this, for me, this is already a sign of biological activity in terms of type 3. So meaning that you have non-white Gaussian correlated noise. And there's really a reason for that. Again, I'll explain in a moment. And along Y, you see somewhat similar, but not so persistent oscillations. But in a way, if you think of where they swim, you see that along X actually they're bounded, the motion is bounded by the cosine here, and it seems they don't prefer to go to the deep sea, while along Y it's unbounded. And that's why you have different oscillations. So it makes perfect sense. Okay, and now we can construct our model, as simple as that. Meaning you take the overdamped Langevin equation and you plug in your distribution of the velocities and you plug in the correlations. And that's what we did, modulo. We amended this model by boundary conditions, meaning we put the turtle into an asymmetric harmonic potential because, as I've just said, here there's the coastline and here's the deep sea. So we modeled this by a harmonic potential with different coefficients. But that seems to do the job very well. You do the simulations, and here you have some sample trajectories. So you have a bunch of them, but you see kind of loops. So I was actually pretty happy compared, just only qualitatively compared with that. And then you can compute the mean square displacement, both from data and from the simulations. And what you see here is really okay. You have the experiment, blue dots. Then you have the simulations. This cannot really be seen, but it matches the data pretty well. And actually the green dots are simulation results from individual runs. So you see that really the experimental data goes very much straight through, so that we are really confident that our our model reproduces the data very well. And now, as this is about anomalous diffusion, well, first of all, this generates loops. Secondly, you see that on shorter time scales, you have actually super diffusion. So you have anomalous diffusion with an exponent of about 1.5, and again, quite well reproduced by the simulations. And again, that makes a lot of sense. Okay, as you will see, and then there's a crossover to subdiffusion because then the turtle hits the boundaries, so it doesn't diffuse any further. Yep.
Just a thought: is it clear that turtles are in the overdamped regime? I mean, it's like
it's
a big object. So.
Good question. Of course not. This is just of what we model here. Of course, ideally we want to go beyond, and I know how. But I mean, this turned out to be so non-trivial that it kept us busy all the time to understand it, even on the overdamped level. But ideally, of course, you want to construct an underdamped model. I hope it will be done sometime.
Because I was thinking, because I see some very sharp corners in the simulation.
Of course.
And in the experiment, I mean, the turtle needs some time.
Of course. No, this is a Mickey Mouse model, obviously, right? I mean, this is the first take, but I mean, as you see, it's quite easy to be constructed from the data, and you already learn a lot. But of course, the full story, and wait to the end, I will propose an underdamped model that should tell the whole story. But this is the first take on it. Okay. And yeah, so at least you learn something. There's some message. The message was first, this loopy motion is not induced by the ocean currents, as you might think. There's work by, how do you pronounce him correctly, Lusky or Lushi? No, not Lushi. Lusky, thank you. Who has actually studied leatherback turtles. And there he argued that they swim with the ocean currents. But loggerheads are different, and then indeed even he says that they perform more complex motion patterns. And indeed here you can put a tracer and a simulation into the ocean, and you indeed see that the tracer moves completely differently from the turtles. So on the other hand, quality is really bad. Here, this is the chlorophyll gradient at the coastline, and the turtles do swim to some extent parallel to these profiles of the chlorophyll, which we haven't built in explicitly into our model. So there's definitely a coupling to the environment, but not the trivial one with respect to the ocean currents. And the chlorophyll is in turn proxy for the food sources. Okay, now back to the looping. When I saw this, I had the hypothesis that perhaps you see these loops are actually pretty big, you know, and I thought perhaps they are that big that approximately the turtle really decides to cover the whole distance along X of the foraging region. So this was, the hypothesis was that the turtles oscillate across the whole foraging region, bounded by the coastline and the deep sea, and that this then gives the looping, so they try to forage efficiently. And now what we did was, or what one can do is, you look at the correlation decay, you identify the maxima minima, you calculate the half period. So if this is true, they should swim over the whole distance by a half period of the oscillation, right? And then actually you can extract the distance they swam during this time from the mean square displacement, and then you can do some matching. And that's what we did. So actually here, if you identify this foraging region approximately like that, you see it's not straightforward. You get 200 kilometers, so it's a lot. You see they swim over large distances. The half period is 410 hours. And if you calculate the distance swam from MSD, it's about 83 kilometers. So you see, I mean, it's certainly not the perfect matching, but at least it's not orders of magnitude away. And you see that actually the loops here are partially a little bit smaller than the whole distance. So this is only a hand-waving argument, but I think there's probably something about it. And I may also say that this depends on the individual turtle. So they all swim a little bit differently, but there is something along these lines, I would say. It's a rough matching. Okay, I need to speed up a little bit. And yeah, I mean, certainly here we have super diffusion, so that may yield a new optimal foraging strategy compared to the famous Levy flights. So you see, you don't need Levy flights to generate anomalous diffusion. Here it just comes from the correlations. Okay, yeah, second example, we have heard about cells, cell chemotaxis. So here you have the 60 neutrophil cells recorded over 30 minutes, 5-second sampling, 360 data points per track. And again, same game, you extract an overdamped Langevin equation. Meaning you need, first of all, the PDF, but here we found that it's rather a simple Gaussian. And then you need the correlation function. And indeed, the correlation function is shown here. This is the data, and the black lines are the fits. And we found that indeed the correlation functions are both power laws along X and Y. But along Y, it's actually pure power law, actually in the overdamped regime here, which is what we applied, you get fractional Brownian motion. While along X, it is tempered. So you have here a kind of power law truncation. So this is now then our model. So overdamped Langevin equation again, we use sample via this correlation decay along X and Y, again from Gaussians. And in a way, I mean, this is of course, compared to the model that you presented, this is of course totally different in a way. It's just based on stochastic modeling. But it seems to do a good job in explaining the data. So again, you see the fits here. So it's constructed from data. And yeah, I should say here are plotted the first and second moments. Of course, there's a drift along the y-axis. So that's why you have a drift term here. And here you have this where there's no drift along x. And here there's a second moment, so mean square displacement. And again, in both cases, due to the correlations, you have obviously anomalous diffusion, both along x and y, with exponents here along y, you have 1.5, and along x, because of the tempering, it varies a little bit. But again, here we have an example of anomalous diffusion, and again, biological activity in terms of model active Brownian particle model 3, because you have non-white noise. Third example, bumblebees. So I mentioned the experiment already. And what was done was that the bumblebees were foraging in 3 phases. namely first spider-free foraging, so they just got nectar here on these flowers. Second, on a predation risk, so they randomly put 4 spiders into some of the flowers, and the bumblebees learn to avoid spider-infected flowers. And third, memory test 1 day later, again with the spiders, just to check whether the bumblebee still remembered to avoid the spider flowers. And first, the PDF. So this is the velocity distribution along y, so meaning horizontally parallel to the wall. And this is the semi-log plot. This is the data, the black dots. And you see we try to fit with exponential, power law, and the mixture of 2 Gaussians. And the mixture of 2 Gaussians definitely works best. And that has a biological reason, because it turns out that there are 2 different flight modes. So again, spatial temporal intermittency. Close to the flower, they move with the small Gaussian. Far away, they move with the large Gaussian. Now, big surprise, no change in the velocity distributions over phases 1, 2, 3. Just exactly the same, which puzzled us a lot, you know. So that means they didn't react to the spider threat by changing their velocity distributions. Instead, they changed their velocity correlation functions, as you see here. So the red one is for phase 1 without the spiders, and you see they move persistently in one direction because they don't feel threatened. So they approach the flower right away, which makes sense. But if they're spiders, they're getting anxious, and then you see the correlations become anti-persistent, so they make turns. That's what it means. And of course it makes sense, because they're trying to avoid the flowers with the spiders. So again, biological activity, non-trivial correlation functions of type 3 in the correlation decay, if you wanted to model that as an overdamped Langevin equation. And simply plug in the mixed Gaussian ones and these correlations, and you're done, at least on this level. Okay, so far to the data analysis. I have 8 minutes left, but I hope that's enough to do a little bit of theory again. Back to the theory. Now, Cartesian versus co-moving frame. How do you make a step? Ask yourself. I mean, I make a step by choosing a certain direction and a certain step length, right? I do not compute my step in the Cartesian frame here in the lecture hall by calculating the x component, the y component, and then I sum up the squares, I take the square root. At least I don't do that. I work in the co-moving frame, and I strongly suspect that any organism is doing as well. And that's an important message in my view. So I think the organisms produce their movements in the co-moving frame, not in the Cartesian frame. But now think of how we are typically modeling a stochastic process, right? We're doing it in a Cartesian frame for very good reasons. A, it's way simpler to write down a process in a Cartesian frame. B, if you're a physicist like myself, that's what you need to do, right? Tracer particle in a fluid, so you do choose the Cartesian frame because the fluid is there and the particle is moving in this frame. But I would argue for an organism, do something different, work in the co-moving frame. It makes much more sense because the noise that drives the organism should happen in the co-moving frame, not in the Cartesian frame. And indeed, go back to Pearson, that's exactly what he did. I didn't tell you, but obviously he did formulate this random walk in the co-moving frame, not in the Cartesian frame, for good reason. You know? So it was there in Pearson's work already. And this has been generalized in movement ecology as a correlated random walk. Same thing in terms of step length and a turning angle. And you simply choose the turning angle from a non-uniform distribution by which you model persistence, which is what organisms do, right? They move persistently in one direction. And this is a correlated random walk, still Markov, but persistence in terms of sampling from, say, rep Gaussian. Okay? So a generalization of Pearson in a straightforward way, if you wish. Now think of a Cartesian representation of a correlated random walk, polar coordinates, as simple as that. So you simply represent the velocities in polar coordinates, speed, s, and orientation, beta. But here there's something that I think people have not generally spotted. Namely, this correlated random walk is exactly identical to active body motion type 2. That's what it is. No? And I think people didn't know because one comes from movement ecology, the other from active body particles. So in a way, they reinvented the correlated random walk, but it's exactly the same. You know, I mean, because remember, you sample the turning angle beta from a Gaussian. This is a Wiener process, so it's the same. Okay, now this is interesting. I think there in the co-moving frame, active body motion 2 is Markov, obviously. While in the Cartesian frame, actually this I learned from Satya, a paper with Basu 2018, that indeed it's non-Markov, which is also interesting. So that, but we know that, right? That you can representations of a stochastic process that is Markov in some frame or coordinates and non-Markov in others. Here we go. And perhaps this is also a biological reason why you should work in the Coulomb frame, because it's somewhat simpler, right? But you do have correlations then if you measure it in a Cartesian frame. Okay, and now here's my proposal, in a way addressing your question about the underdamped dynamics. My proposal is that it should be very promising to model biological movements by an underdamped generalized Langevin equation in the co-moving frame, which looks exactly like that. Namely, for the two variables, the turning angle alpha and the speed s, and here you see they're both underdamped. You have a drift term and now a diffusion term, meaning noise, which can be arbitrarily correlated, arbitrary distributions to be extracted from data. And indeed, we put forward this equation already. You see it more than 10 years ago in this paper. We just wrote it down and applied it to data analysis. It fell from heaven. Yes, I know. But I really think it deserves more attention for data analysis. And that's what I said. And now the nice thing is that this combines all three types of active bounding particles. So if the drift terms include velocity, then there you have ABM1. If you have non-uniform turning angles, you have 2. If you have correlations, you have type 3. So you have all there. And even more, you get correlated random walk as a special case, Levy walks, sample from Levy step length distribution, you get Levy walks. And of course, random walks are in there as well. And if you go to the overdamped case, again, then you get that. And I think you can also model run and tumble if you wish. Okay, 2 examples of applications, and then I'm done. First, we actually constructed this underdamped equation for Bumblebee flights. So here we just looked at them far away from the flowers. And making a longer story short, you can do data analysis, and what you get is, for the turning angle, this overdamped equation, where you sample from Gaussian, which however is polar correlated. So here you have already active bounding motion type 2 and 3. And then you can extract the speed equation, and you see we get a non-trivial friction term, which is like active bounding motion type 1. And we get again anti-correlated Gaussian noise, so we have also type 3. And this is our model for the bumblebees, and it works surprisingly well for reproducing the data. We were quite positively surprised. So this can be done that you apply these equations to data, you can extract everything from the data, you have a data-driven model. And you have all 3 types of active motion, which I find quite nice. Now, if you're a theorist, then I want to motivate a little bit why it may be very promising to check out stochastic dynamics in the co-moving frame. There's a recent experiment by my colleagues, Alessia Gentili and Giorgio Volpi, where actually they were talking about that last year, where they drive a colloidal particle by magnetic fields. And they can do so by telling them by which step lengths to move and by which turning angles. So if you give them the distributions of step lengths and turning angle and the correlations, they can implement that in an experiment. And they asked me the question, okay, we want to generate Levy walks. Okay, this is easy. We know what to do. But we also want to generate friction bounding motion. And then Giorgio told me, tell me of how I formulate friction bounding motion in the co-moving frame. And I told him, I have no idea, and I don't even think it's possible. And if any one of you knows, let me know, but I don't think it works. Because it's obviously non-Markovian, and if you want to transform a non-Markovian process from Cartesian into the co-moving frame, you're in trouble. And this is already known for Levy walks. Namely, there's a work by Vasily Savodaev that actually 3 different Levy walks, 2 defined in the Cartesian frame, 1 in the co-moving frame, and they're all not identical. So you see, there's something interesting going on, meaning it depends. You may get different stochastic processes, whether you define them in the Cartesian frame or in the co-moving frame, and they cannot be transformed into each other. And I suppose the same is going on for FBM, but so far open question. Okay, 50 seconds left. Okay, almost. Then last slide is ongoing work with Norberto, who's sitting over there. Namely, what we tried to do was to transform, now as an example, Ornstein-Uhlenbeck from Cartesian into co-moving frame. And this can be done, because this is obviously Markov. So as I'm running out of time, just flashing through. So first, you formulate the velocities in polar coordinates. Then, obviously, you have an Ito-Stratonovich problem. So here we apply Ito, and this is known. You find it in the Gardiner's book. You calculate the equations for the orientation and for the speed. You get these ones with Gaussian white noises in both cases. But now you want to have actually a turning angle equation for this, not in terms of the polar angle beta. And this is actually what we're still working on. I mean, you presented results to me 1 hour ago, so therefore I don't flash an equation. But it seems that the PDF for the turning angle is a kind of given by Bessel functions. It looks like a wrapped Gaussian, but at the moment it seems you're getting something different. So this is work in progress. But the bottom line is that we are very sure that we can exactly transform an Ornstein-Uhlenbeck process into the Kumui frame two equations like that. So one for the turning angle and one for the speed, where you sample from an exponential correlated Rayleigh for the speed and from this kind of Bessel stuff Markov for the turning angle. So this can be done. So you can formulate equations like that. You can transform them from Cartesian to Kumui frame. But I think if you're non-Markov, you're doomed. I don't think it will work. But you get an intuition of how the equations look like. Okay, summary, here we go. What I tried to present was, or what I'm proposing is a fusion of movement ecology with active Brownian particles and anomalous diffusion as a model for modeling the movements of organisms. This is this underdamped Langevin equation in the Kumui frame that I put on. I think that could be very promising to understand movements of organisms, perhaps even on all scales to some extent. I'm not saying this is everything, of course not, but at least for understanding movements, I mean, I think it's promising and deserves some attention. And certainly we know that Langevin dynamics is generally a powerful paradigm in many fields of science, physics, climate, finance. It has been applied, so why not applying it to organisms? It's there. Okay, and as hopefully I've shown to you, this generalized Langevin equation that I'm putting forward is amenable to experimental analysis. You can extract it from data, and theoretically it seems quite interesting, this interplay between Cartesian and Kumui. And it includes all kinds of biological activity and anomalous diffusion. So it's all in there. Okay, that's it. 47 seconds over, but okay, I barely made it. So this is just acknowledgement to all colleagues who contributed to this work. You see there's some work in progress. And in fact, Luca, this is exactly the book chapter I'm writing for your proceedings. So it's written, will be submitted soon, and I'm planning to write another short paper. Thank you very much for your attention.